It's my pleasure to welcome today Nate Sanders, who is one of the candidates for the directorship of the Biological Station. Nate is currently Professor and Associate Dean for Academic Affairs and Faculty Development in the Rubenstein School of Environment and Natural Resources at the University of Vermont. Earlier, he got his PhD at Stanford. He was a postdoc at the University of Tennessee, and then faculty at the University of Tennessee for 10 years. And after that, he moved to Denmark and was a professor and head of the biodiversity section at the University of Copenhagen before starting this position at the University of Vermont in 2017. His research encompasses community ecology, ecosystem ecology, and macroecology. And he studies the organisms that are the most important critters that run everything, the ants, plants, and soil microbes, at least in terrestrial ecosystems. And so, and he uses this system to address the effects of global changes on biodiversity. So without further ado, let's hear about macroecology, biodiversity, and biological stations.
Hi, everybody. Thanks very much. There are a couple seats up here, one there.
Yeah, please come on in.
One here.
Two here.
So while everyone's coming in, one of my favorite things about seminar series is that, much like students in a large lecture hall, everyone always sits in the same place when they go to a seminar. But this is a relatively new room, so I don't think everyone has figured out where they're going to sit yet. When I was at the University of Tennessee, everyone sat in exactly the same chair for every seminar. And I recently saw a picture posted on Twitter of last Friday's seminar, and sure enough, there is Lou Gross sitting in the front right and Randy Small sitting in the front left. So thanks again for the opportunity to come talk with you a little bit about my research, a little bit about how I view biological stations. Annette is leaving. I've already offended her. And a little bit about how I see those two things overlapping, that is my research program and biostations. The work I'm going to talk about is work that we've been involved in since about 2007. And really the work is work done by a whole host of postdocs, collaborators, grad students, and undergraduate students. What I hope to convey in this talk is my general approach to doing science, the kinds of questions I ask, how I take advantage of the little things that run the world, the ants, to ask pressing questions in ecology, and then also blend in how I see my research at a biological station and how, more importantly, I see facilitating research and teaching at biological stations. Since I've been giving talks like this, I always start with this slide. This is the slide that motivates the research we do in my group. Biodiversity is distributed unevenly on the planet, and there are, as you know, numerous global change drivers that are changing where that biodiversity is, how people rely on that biodiversity, and how that biodiversity influences how ecosystems function. The causes and consequences of biodiversity change from meter square quadrats to entire continents is where our research is. Now, for me, the cool thing is that field stations are where we, that is mostly I, study biodiversity. This is sort of a pop quiz. I'm going to show you some field stations. Where is this one? Anybody know? Chiricahuas, yes, the Southwestern Research Station. But it's field stations like this where we study biodiversity. Quickly. Jasper Ridge Biological Preserve. Field stations curate biodiversity data. Jasper Ridge is famous for curating a long database on the population dynamics of the bay checker spot butterfly until it went locally extinct. But it's this curation of these long-term data sets that allow us to explore how biodiversity is changing in response to global change drivers. Field stations are where we introduce students to biodiversity. No one will know this one, I predict.
Your house.
Close. This is the Department of Ecology and Evolutionary Biology field station adjacent to Great Smoky Mountains National Park. And so this is my community ecology class where I was introducing them to Robert Whitaker's work in Great Smoky Mountains National Park as part of a community ecology class. As you know, field stations are incredible places where we introduce students to the organisms that actually occur in nature, how those organisms interact with one another, how they interact with their environment, And hopefully we use that information about organismal biology to introduce students how to ask and address important and pressing questions. Field stations preserve biodiversity and engage the public with it. Yes, lots of people know this one. This is the Rocky Mountain Biological Lab. It's been there since 1928. Some of these cabins have been there since 1928 as well. The Rocky Mountain Biological Lab does an incredible job of engaging the public in the science that happens at the Rocky Mountain Biological Lab and environs around it. There are groups of retirees who spend days walking around learning about butterflies. There are K through 12 students who spend 2 or 3 days a week in nature camp. There are high school students who spend 2 days a week learning everything from fern biology to how to take a soil core and understand columbian abundance. They've done an incredible job not only of doing science, but also engaging the public with that science. Now, I think that many field stations over time have developed reputations for what they're good at, and they've lived on those traditions. I think that some of the trade-offs that you know about, these are the same trade-offs that, or assumed trade-offs that perpetuate much of higher education, that you can be really good at teaching, but if you're really good at teaching, you can't be good at research. Or if you're really good at research, you can't be good at teaching. I've heard that my entire life. Of course, it's not true. There are also trade-offs such that you can invest in understanding organismal biology, who those organisms are, what they're called, what their evolutionary relationships are, what their life histories are. But you do that at the expense of asking and addressing pressing questions in ecology. That's a presumed trade-off. Of course, it's not true. It seems to me that there's this trade-off that exists, especially in higher education, that if you engage with the public, with K-12 students, with students from urban high schools, with retirees, with anyone else, that you do that at the expense of what the ivory tower values, that is research dollars and publications. Of course, that's a presumed trade-off. It doesn't need to exist. There are other trade-offs about whether if you invest in understanding a place deeply so that it's in your bones, that you intuit everything about the organisms and how they interact, you do that at the expense of understanding anything about global scale ecology. That's a presumed trade-off. Of course, it doesn't exist. Field stations have been instrumental in breaking these trade-offs. Some have invested heavily in teaching at the expense of research. Others have invested heavily in organismal biology at the expense of addressing pressing questions, but the best field stations break these trade-offs. My vision for the field station for the University of Michigan Biological Station is to break these trade-offs, to engage in high-impact research and high-impact teaching, to understand organismal biology, but also ask and address pressing questions and ensure students know how to do that, to engage the public, but also engage in high-impact research, and to do place-based ecology that matters at global scales. And so what I want to do in this talk is show you a little bit about what I've been doing in my own research program that I think breaks some of these trade-offs. And then in the end, I'll focus on some of the things I'd like to do at the station that wrap up these trade-offs. So for a long time, I've been interested in a variety of issues around species invasions. The first organisms that I studied in the field was the Argentine ant. The Argentine ant, as its name suggests, is from South America. It's been spread all over the world. I invested heavily, truly, blood, sweat, and tears, and my increasing susceptibility to poison oak to study the Argentine ant at Jasper Ridge Biological Preserve. For a month, 3 times a year, I scoured the entire 480-hectare preserve looking for these ants. And I documented changes in their distribution and changes in their impact over time. We've scaled that work up to try and understand what the global scale impacts of this ant are, how climate and native communities might resist invasion by Argentine ants. And this is work led by Nuria Pascal, a colleague in Spain now. And it's the sort of example that I really like because it started with focusing on the impacts and spread of a particular species at a particular location but we've scaled it globally to understand whether what we learned at a particular place over many years scales to the globe. And the answer is yes. I really wanted to do an experiment. I am an experimental ecologist, as you'll see, with these ants to try and understand whether biotic resistance, that is the structure of these local ant communities, climate, and resource availability influenced invasion dynamics in this species. I wrote a paper about the spread of this ant in Jasper Ridge Biological Preserve, and here's something to never say in an interview. That paper was rejected and never published because it was entirely correlational. There was no experiment. And we couldn't think of how to do the experiment with the ants. So at the University of Tennessee, we set up an experiment in beautiful 3 meter by 3 meter plots where we manipulated biotic resistance, we manipulated propagule pressure, we manipulated resource availability to understand how those characteristics of these communities influenced invasion by Lespedeza cuneata, a plant. And so the reason I tell you that story, even though I am an ant-ecologist, sometimes you can't ask the questions you want to ask with the world's coolest organisms, and you have to work on plants. That's another thing never to say in an interview, by the way. But it shows my approach to ecology. I, of course, am tied to organisms, but I also want to ask the questions that need to be asked in the systems that allow me to ask them. Another example is a plant insect example. I spent a lot of time watching ants interacting with one another in the desert, and I came to understand that there was variation among individuals and how those colonies interacted with one another. There's variation among individuals within populations and within species. You can look around this room and see that there's variation among individuals within the same species. And so the question is, does that variation matter for biodiversity in the next trophic level above, in the next trophic and below ground? And does that biodiversity influence ecosystem function? And so in a series of experiments where we manipulated genotypic diversity, the number of genotypes in populations of goldenrod, we found that, sure enough, the number of plant genotypes in a population influences above ground net primary productivity, which in turn influences herbivore richness, which in turn influences predator richness. And so what this work shows is that the loss of variation, even within species, has impacts on biodiversity and other trophic levels, just like the loss of species from communities does. I've also branched out to work with colleagues on the Tibetan Plateau in China, where we've tried to understand how the loss of biodiversity in multiple dimensions above ground and below ground influences the suite of functions that ecosystems provide. And so this is just an ecosystem multifunctionality index with a variety of different biodiversity for a different suite of taxa. And we've shown how the loss of biodiversity is related to the loss of multiple ecosystem functions. At another site on the Tibetan Plateau, we've taken advantage of long-term data sets going back to 1980 that look at how soil moisture has declined through time, how ANPP has varied through time, and how the relative abundances of grasses and sedges has changed through time, to try and understand why that's occurring. So we've taken advantage of long-term data and overlain an experiment to try and understand how biomass shifts above ground to below ground in response to this decrease in soil moisture. Again, it's the sort of approach that I like to take, where you have a long-term observation or a macro scale observation, and to understand the mechanisms, you do a local experiment. Similarly, we've worked at tree lines in Colorado to try and understand how a suite of ecosystem parameters vary at tree line. And so here's our site in Colorado. And then we branched out to ask the same questions measured in the same ways at 7 other sites around the world. What we're trying to do is know whether what we understand in one place is common to the rest of the world. The answer here is yes. At the Rocky Mountain Biological Lab, again, we worked on interaction networks to understand what happens either with phenological shifts or the loss of species. Are interactions rewired, or does species turnover occur? But all of that is really about my own work on the ants. And so the taxon that I think about the most, dream about the most, work on the most are these organisms here. And the kinds of things we've done are motivated by lots of questions, lots of observations, and because everyone has experience with ants. Has anyone experienced this sort of interaction with an ant? A handful have, okay. This is the other thing I always do in talks because I've been taking data on this for like 12 years. How many people have seen this movie? That is slightly more and slightly younger than the people who usually raise their hands for this.
These really big ones?
They are the really big ones. Yeah, okay, there you go. Long time ago, like 1954. Yeah. So this is a late night movie. You should all run out and watch it. It's, now it's really, really funny. But radioactive waste spills in the desert in Nevada. Everyone knows if you pour radioactive waste in the desert in Nevada, the ants grow to be the size of, SUVs, and they go around eating people. So much younger hand raising that time than most places. I don't know what that says. But ants have inspired movies, all these sorts of things. They're also incredibly, incredibly abundant. This is a schematic by my former postdoc and friend Rob Dunn, where they estimated the number of ants in Manhattan. So this big black circle here is the number of ants crawling around in Manhattan. That number is 1.23 billion ants crawling around. In Manhattan, there are 1.6 million people crawling around. So the sizes of these circles show the relative abundance of ants to people in the most densely populated city in the United States. And this is a city. So imagine what it must be like at UMBS. The number of ants greatly outweighs the number of people there as well. They're incredibly abundant, and because of that abundance, because of their suite of life histories, they have huge impacts on ecosystems. This is my favorite ant. This is an ant that occurs in woodlands around here. It's a Phaenogaster rudus or Picea. It's a keystone seed disperser. Many understory plant species, by some estimates more than 50% of understory plant species have their seeds dispersed by this ant. Not exactly this ant, but a species of this ant. And so the ants rip off this elizome and disperse the seed somewhere. But again, these are important mutualisms in forests around here. Ants are important herbivores. They're important predators. They're mutualists with plants. In some places around the world, and even here, colonies can be gigantic. This is a redwood ant, Formica rufa. There's a former postdoc, Israel del Toro, who's actually taught at the UMBS before. And they have lots of effects on soil nutrient dynamics. And so here's a colony of another species of Formica, and you see this ring of fertilized grass around the nest. And so for those reasons and many, many more, I would love to give a talk entirely about how cool ants are. It's probably not appropriate for a talk like this, but at some point, I hope you hear me give it. But for those reasons and many, many more, I am interested in what governs the distribution and abundance and biodiversity of ants spatially and temporally, and how changes in that biodiversity affect how ecosystems work. We started doing this work in Great Smoky Mountains National Park in around 2007 in forests like this, where a team of undergrads, grad students, and I collected ants in 29 sites along the elevational gradient, mostly in the leaf litter. So the way this works is that you scoop up the leaf litter into this sifter, and the ants and detritus fall down into the bottom of this, and then you put it in this little black pillowcase. You bring it back to the lab, and what happens next is you put it in these Winkler sacks, and these are sort of mesh sacks. The litter dries out, and the ants fall down into these vats of alcohol, and then you sort through the ants. So we did this over about 3 or 4 summers with these students, mostly hiking kilograms of leaf litter around in backpacks. We sorted these ants for about 2 years, and then at the end of the day, we were left with this, a scatter plot showing that the number of species in a community declines with elevation. Now in 2007, you could stop there and say, great, we've documented an elevational diversity gradient. Diversity declines with elevation. But of course you want to know why that is. I want to know why that is. Well, it turns out that, as you likely know, abundance of almost any organism tracks temperature. And so this is mean annual temperature of these sites. And this is an estimate of the number of colonies at those sites. And as temperature increases, so does abundance. Warmer places have more ant nests than cooler places. Again, not so surprising. Because of that, the number of species in a local community tracks abundance. Places where there are more individuals have more species. That could simply be driven by a sampling effect. If you have more individuals or more marbles in a jar, the more marbles you draw out, the more likely you are to get more different marbles. It could also be ecology. There's something that leads to increases in abundance in some places, but lower abundance in other places. So all of that is to say that the number of species in a local community tracks mean annual temperature. As temperature increases, there are more species in local communities. This is generally true for lots of taxa. There are more species in local communities in Costa Rica than there are in Canada. There are more species in Connecticut than there are in the Yukon Territory. Warmer places tend to have more species than do colder places. Now, this work is entirely observational, entirely correlational. It was a lot of work still, but it's all in one place along these elevational gradients in Great Smoky Mountains National Park. And so what we wanted to know next is what we learned at this particular place, something that informs patterns at global scales. And so the way I did that was to give a talk at a national meeting and say, here's a pattern we've seen in Great Smoky Mountains National Park. And to the audience, I said, if anyone would like to collaborate on a global synthesis where we all compile our data sets to ask whether this pattern happens globally, meet me at that bar and I'll buy the first round. And so all of these people showed up. I bought everyone a beer, not using federal grant dollars, for the record. I'm obligated to say that. But what it meant was that for about, $297, we got a data set of 4,000 ant communities. So these are local places where an ant biologist, an ant ecologist has gone out and sampled that community, identified all the species, and for many of them, estimated abundance as well. And so what we've done with these data are things like this. And so this is a recent paper by Javier Arnan, where if you just look at this graph first, this is the natural log transformed species richness of these sites as a function of temperature. And so as temperature increases globally, so does species richness. Of course, there is variation. There are some places that are hot with very few species. There are some places that are colder with just as many species as there are in warm places. But overall, species richness will increase with temperature. We've also done pretty interesting things, again with Javi Arnan and a master's student, looking at how invasive species disrupt these macroecological patterns. So again, temperature leads to increases in species density, but the presence of invasive species disrupts or changes that global scale macroecological pattern. So that the relationship between local species richness and temperature no longer exists once invaders show up. So all of that, I'll say off the record, I'm now at the stage of the interview where I'm starting to lose my voice. So all of that is to say that temperature matters. And if I stopped there, no one would care. But we want to know why temperature matters. What is it about warm places that lead to more ants? What is it about cold places that lead to fewer ants? Of course, it's something about the physiology of the organisms, right? They are insects, they are ectotherms. Temperature matters. And so what we did next was, do you know when you make creme brulee and you have those little torches? I forget what they're called. So we actually didn't do that, but I love showing this slide because there's always someone in the audience who either, squirms or it's more scary when no one reacts at all, and they just say, of course that's what they did. But the real reason I show this slide is to make what we actually did seem not so bad. What we actually did was use water baths to raise the temperature and lower the temperature of the ants to estimate their thermal tolerances, where CT max is the maximum temperature an ant can tolerate before it loses its ability to right itself. CT min is the minimum temperature it can tolerate before it loses the ability to right itself. That is, get up when you shake it in a little vial. And so here are the results. What this shows on the x-axis is the mean annual temperature at the site at which we collected these ants from the field in Great Smoky Mountains National Park. The y-axis is CT max, that is the maximum temperature the ants can tolerate before they lose the ability to right themselves. And each of these different clusters of color is a different species. And so what you see is that some species have low CT max, some species have high CT max. Those ants that have high CT max can tolerate higher temperatures than those ants that have low CT max. Some ants have low CT mins, that is, they can tolerate low temperatures. Other ants have high CT mins. They can't tolerate low temperatures. Interestingly, this ant here that can't tolerate high temperatures but can tolerate low temperatures is called the winter ant, Prenolepis imparis. It occurs around here. You'll see it out today. It's the only ant out foraging, probably. The other cool thing about these patterns, as you likely see, is that there's variation within species as well. So depending on where you're collected, where along the elevational gradient or temperature gradient you're collected, your CT max is either higher at a warm place or lower at a cold place. Similarly, if you're the winter ant and you're collected at a warm place, your CT min is higher than if you're collected at a colder place. That is, ants from cold places can tolerate cold temperatures, ants from warm places can tolerate warmer temperatures, and then the converse is true. Now, Lacey Chick, a former PhD student of mine, repeated this work across the temperature gradient from North Florida all the way to Maine. And this is just within a single species, Aphenogaster rudus, and she found the same thing across the latitudinal gradient. CT max tracks the temperature of the site, and so does CT min. So this is really cool. It suggests that, that there's local adaptation going on, or it suggests plasticity in these populations, or individuals in these populations. So to test that, we brought ants back to the lab and reared colonies at different temperatures. We reared them at 20 degrees C, which is on the low end of the temperature they would experience in the summer, and 26 C, which is on the high end, and then measured the CT max of workers from those colonies reared at different temperatures. And if there were acclimation going on, you would expect that if you reared colonies at warmer temperatures, their CT max would be higher than if you reared them at lower temperatures. And you don't see that for CT max, and we don't see that for CT min. Again, what it suggests is that there's local adaptation in these populations to the temperatures they experience. Everything I've told you so far does not include an experiment except when we drop ants into water baths. We've just measured where ants are, counted them, driven from Florida to Maine, counted them, collected them, thrown them in some vials. That's it. What we wanted to do next was an experiment where we manipulate temperature. And we did these experiments at Duke Forest and at Harvard Forest. And we chose these two sites in part because they're field stations, but also in part because Duke Forest is the southern range boundary of about 25 species in the eastern US, and Harvard Forest is the northern range limit for about 25 of those same species. And we wanted to know whether warming affected populations at low elevations differently than at or low latitudes in southern range boundaries than at high latitudes in northern range boundaries. So this is work with an incredible team. This project was initially a DOE-funded project, and then a NSF Dimensions Biodiversity project. And the way we did it was with these open-top chambers where we're pumping warm air into these open-top chambers, and then ant queens can fly in, ant workers can crawl under, And then these plots are big enough for entire communities of ants to live their entire lives and interact with their neighbors and their environment. And here's a thermal image of one of these chambers. This is a control chamber, just showing you what it looks like, and then a warm chamber, where again, we're pumping warm air into these plena with holes poked in it, and the warm air is distributed across the entire chamber. This is what those plena look like. And so that's where the warm air comes in, moves around these plena, and then we have a series of pitfall traps, take a variety of soil cores, measure almost everything you can measure in response to these warming treatments, but I'll focus only on the ants today. This experiment is set up in a regression design at both sites, where there are 3 control chambers, and then chambers are warmed in half-degree steps, all the way up to about 5.5 or 6 degrees above ambient. So whatever the temperature is outside, the warmest chamber is always 5.5 degrees above that, and the 2.5 degree chamber is always 2.5 degrees above ambient. And so what did we learn? Well, the first thing we learned, and this is work by Sarah Diamond, a former postdoc on the project, was that the thermal tolerances of the ants themselves predicted where they occurred in these experiments. And so here, each dot is a species, and what this graph shows is that those ants that can tolerate warmer temperatures, that is, they have higher CT maxes, were most abundant in those chambers that were warmed above ambient. So this is the temperature of the chamber where they were most active. So again, ants with high CT max are most active in warm chambers. This functional trait, that is thermal tolerance, predicts how species respond to warming. At a community level, what we learned is that with experimental increases in temperature, the ordinal date of peak abundance, that is the total number of ants out crawling around, was shifted, such that under ambient conditions, the ants were most active, and this date is averaged across multiple years. The average date of highest activity was July 19th. But as we increase temperatures experimentally, what we see is that the ants are more active earlier in the year. And this likely has important consequences for phenological mismatch between the ants and the seeds they disperse. In the lab, we brought this ant, Aphaenogaster rudus, the important seed dispersal, into the lab and then reared colonies at different temperatures. And what you see on this survivorship curve is the percent of colonies surviving up to 80 days, reared under different temperatures. And you see that colonies reared under the highest temperatures over time have the lowest survivorship relative to control chambers. So there are direct effects of temperature on colony survivorship. In the field, we learned that temperature affects colony size. So in warm chambers, colonies are much smaller than in ambient chambers. So as it gets warmer, colony size shrinks. That in turn has likely effects on the fitness of these colonies. And so if you look at colony size for those that don't produce reproductive ants relative to those that do, you see that colonies that are producing reproductives tend to be bigger. And as colony size increases, the number of reproductives produced increases. If you think of these like plants, those plants that are bigger produce seeds. As plants get bigger, they produce more seeds. And again, all of that is driven by the direct effects of temperature on colonies. One of the things we did was put out these artificial nests in these chambers, and then every month we would go see who occupied the nest. And at Duke Forest and Harvard Forest, we looked at several components of population dynamics. What this shows is a schematic where blue arrows show negative effects, red arrows show positive effects. MAT is mean annual temperature, and then these boxes are different species. And so this means that temperature had a strong positive effect on this ant, Chromatogaster lineolata, a negative effect on this ant. This invasive ant had a strong negative effect on this ant. And these metrics are occupancy, who's occupying the nest, colonization, who moves into the nest, and extinction, who moves out of the nest. And so what you see is that interactions among these species for limiting resources mediate the effect of temperature. So again, there are negative effects, but these negative effects can occur because of positive effects on particular species. So warming and interactions interact to shape population dynamics. One of my favorite graphs is this one from Katie Stubel, who's a former PhD student. And Katie went out every night for several weeks and just for 24 hours a day and just asked whether the ants were out foraging. So again, this is Aphaenogaster rudus. This is the ant Chromatogaster lineolata. This ant is the important seed disperser. This ant doesn't do anything except mess with other ants. Under ambient conditions, Aphaenogaster rudus spends about 10 hours a day foraging. Chromatogaster spends about 7 hours a day foraging. But then once we turn up the dial, you see that that Aphaenogaster rudus essentially shuts down, but Chromatogaster lineolata drinks 7 Red Bulls and just forages 24 hours a day. And so again, this is the ant that is dispersing seeds. This is the ant that isn't. And so there are likely consequences for those plants that rely on these ants to disperse their seeds. Now, one of the things that I hope to convey in that section of the talk is my approach to doing science. And it's the same approach I would take at the biostation. I am interested in organismal biology, but I use these organisms to ask pressing and important questions about how functional traits might predict how species respond, how those functional traits might be related to distributions, how those traits might relate to survivorship and in turn reproduction, and then how all of those are related to how interactions and the environment interact to shape colonies. And then I scale that up to a macroecological perspective. Now, I started with this question, this statement: biodiversity is distributed unevenly on the planet. This walk through some of the work I've done over my career was actually a walk through field stations that I visited. Almost everything I showed you was work done based at a field station, whether it was the EEB department at the University of Tennessee, or the Rocky Mountain Biological Lab, or a NEON site, or Jasper Ridge, or Duke Forest, or Harvard Forest. The kinds of work that I do is based at field stations. The kind of work like this thrives at field stations. But of course, I'm not here to talk about these field stations or NEON. I'm here to talk about this particular place. I also said that at the beginning that field stations are where we study biodiversity. They are where these organisms occur in nature, where we can introduce students to them and what they do. When I envision the University of Michigan Biological Station and people studying that biodiversity and how global change influences biodiversity, I imagine hosting workshops at these places with people from all over the world, where people come to the University of Michigan Biological Station to think about and to synthesize work that others have done and work that's occurred at UMBS. I envision international collaborations. This is work or an experiment at the field station in the Tibetan Plateau, where we have networks of experiments based at UMBS, but also distributed globally. And we bring collaborators from these networks back to UMBS. So not only is work occurring on the ground, but we're synthesizing work from multiple sites around the world. So the research I envision is not just experiments and observations happening at UMBS, but synthesis of work from around the world. I mentioned at the start that field stations are places that curate biodiversity data. There's a long-term record of records of suites of observations from a variety of taxa at UMBS. Field stations curate that data and allow us to ask questions about how biodiversity changes through time. I hope to do that at UMBS, but I also hope to collaborate with people in museums at the University of Michigan and elsewhere. So final pop quiz, what museum is this?
CU.
Boom, the Henderson Museum at CU. I didn't think anyone would get that. So I was an undergrad at this museum, and I worked on the ant collection in this museum. I learned how to identify ants, and this slide is mostly for Tim James. This is the best book I've ever read, The Ants of Colorado, written by the curator of insects at the Henderson Museum. So much of his collection was there. I learned how to identify his collection with his book. This book also had all of the life history and ecological data for 163 ant species that occur in Colorado, 500 or 5,000 occurrences in over 166,000 specimens. I knew these ants. I loved this book. I was driving to my field site at the Southwestern Research Station, and the axle broke on my car in Wickenburg, Arizona, which is not a place you want the axle on your car to break, I can tell you in hindsight. And so I was stuck in a motel in Wickenburg, Arizona, and decided to do something at the time that was relatively uncommon, and that is digitize diversity data from a museum. And this is a figure from the second paper I ever wrote, and it shows the elevational diversity gradient for ants in Colorado, again generated from data in a museum that I knew about. And so while field stations curate biodiversity data, so do museums. And I think the synergies that form between museums and field stations are where exciting questions like this one used to be are where it's at. Here's another example of an exciting question from a museum. This is work that you may have heard about last week, but it's data collected by Emily Meineke, AMA, me, and Jonathan Davies, where Emily went to museums and looked at the probability of herbivory on 4 common species in New England through time, and showed that these herbarium specimens reveal increasing herbivory over the past century. And that's the sort of ecological science that I think that field station biodiversity science and museum scientists can collaborate on. Sorry. I also said that field stations are where we introduce students to biodiversity. And again, this is one of my community ecology classes. And as I was putting this talk together, I was actually a little bit emotional because I know where a lot of these people have ended up, and it's incredibly impressive. Not because of me, of course, but just knowing where they are now is really, really cool. But at UMBS, what can we do to increase involvement of faculty, students, postdocs at UMBS? Is it possible to develop a teaching postdoc program where postdocs are funded to do research here, but also be mentored in teaching field courses at UMBS? I think the answer might be yes. Are there mechanisms in place in other departments that have other field stations, like in Wyoming, that allow people to teach in those places? Yes. I think that the administration here is supportive of that. Are there ways to enhance or create interdisciplinary offerings in, I always say PITE, but I think it's Pete, but I'm going to say PITE. Great. Or other campus partners. And so this is a sustainability course that AMA and I taught in Costa Rica, where these students came from all different backgrounds and basically did the OTS tour and did a suite of service learning projects as well. Are there ways to synergize or sync up or refine parts of the curriculum at UMBS so that it aligns with curricula on campus, so that students are not only encouraged, but realize that it's easy to take classes at UMBS that count toward their majors here? I hope that's possible. This is just a picture to show that I've always worn vests my entire life. And because I see I have like 2 extra minutes, one of the things I always do on these trips on the first night is have the students write community ecology haikus. And so they write them as I make dinner, and then after dinner, they read them. The best haiku was called Nate's Red Vest that year, and I still blush when I mention it. Are there ways to think about working with campus partners to make the University of Michigan Biological Station carbon neutral? As a side note, this is not my vision for what it should look like there, but this is a building at Norwich University, I think, where they are trying to go carbon neutral. Are there ways to do that here? Is there support for that in the administration? After I've been here for a day and a half, the answer seems to be yes. And that's the sort of thing I would like to work toward with your help and with partners on campus. Is it possible to think about graduate level courses for students from around the world? So Amy and I often teach courses internationally. This is a course we taught at Peking University in Beijing. There's me right there. Again, another fun picture to remember this experience, but I think that UMBS could be a location for courses like this in the shoulder season, or if there are enough cabins that are winterized, in the winter as well. I mentioned that field stations preserve biodiversity. They have a conservation ethic or ethos or mission, and then they allow or facilitate public engagement. Are there ways to incorporate citizen scientists from K through 12 all the way to 99 and above at UMBS? Might we think about a docent program if one doesn't exist, where people in neighboring towns and communities learn the natural history and organismal biology and the work happening there so that they can engage with the place and help us engage with the public? Is it possible to think about high school students from Chicago, Detroit, all over Michigan, not just in cities, but also rural schools, again, K through 12, who might visit UMBS? Should their teachers come? Could there be teacher workshops that train teachers how to incorporate natural history, organismal biology, and global change into their curricula? So my vision really is to think about breaking these trade-offs at field stations, but of course at UMBS in particular. Now, achieving this vision requires help, of course. The help that it requires will include things like development and fundraising. I've started to work on this where I am now. I think the University of Michigan is very good at these sorts of things, but there hasn't been development focused on the station from the university, at least I think not enough. So we could use help there. I think that there's a tremendous opportunity for investment from the dean's office. There is a new dean who, based on my interactions, is very enthusiastic to invest in UMBS. I think the opportunity is ripe to do big things there. I know I'm supposed to talk about my vision, but my vision is truly a shared vision. I will, if I'm offered this position, I will not come and say, here's my vision, let's go there. Instead, we will develop a vision that includes not only the people in this room, but other people on campus in other programs, people at other universities, other institutes, and community members around the station. Finally, I think for any of this to happen, we have to have and develop and facilitate an inclusive and welcoming environment at UMBS. That requires serious work. I will admit I don't have a solution for that today, but it's the sort of thing that we all have to work toward together. And with that, I'll thank you and again this incredible group of collaborators. And I think there is time for questions. If you have a question, I have to repeat it on the microphone, so don't be offended. Yeah.
So, briefly, you said you don't have a solution for the inclusive environment, but how would you recommend to get the ball rolling on it?
Yeah, so the question was for everyone who's going to watch this later or on YouTube when it racks up thousands of views. How I don't have a plan now, but how might I get the ball rolling to get a discussion going so that a plan could evolve? I think there are lots of things to do. First, like most things, I and we have to know more about what the situation is like And I don't know enough about the details of what life is like there, but I think that where I have been over the past several years, there's been a focus on these issues as well. I have been lucky enough to be in workshops and trainings that have been led by incredible people that have helped me understand other people's paths, where they come from, what they might be experiencing, and then what we can do. And I think I said this earlier in an interview, I went into those meetings reluctantly because I have typically disliked that kind of forced workshop, but some of them are so good that I think that they can help both the community at UMBS and also here. I also think we could envision ways to facilitate visitors to UMBS, whether they're short-term or long-term, because one of the things that we experience in Vermont, which is one of the widest universities in the widest state, is that when we recruit grad students, faculty, undergraduates, they're reluctant to come because they don't see anyone like them there. And so it is a horrible experience to realize that you have an opportunity to recruit this incredible faculty member or grad student, and they don't come for reasons that I, as an individual, have no control over. The second thing related to that, though, is that recruitment is important, and there are ways to do that, but retention is equally important. Retaining faculty, postdocs, grad students, undergrads is just as important, and there are ways that we've been able to address that include, again, these meetings where people have open and frank discussions about ways forward. That's sort of a long-winded answer, but I think it requires lots of people like you asking these questions and people like me trying to come up with answers, but then listening to everyone else about what has worked and hasn't worked. Yeah.
So the question you kind of mentioned about, field stations sort of being a place where you're doing science, specific, unique to that local location, but then it's like global implications, right? So are there some particular areas, I don't know how deep your knowledge is of the station and the environments there, but are there some particular areas that you could sort of highlight either for your own work or more broadly that you see the station and its location be particularly well suited for?
Yeah, so the question was, are there particular questions or approaches or ideas that the station seems really good for addressing, and how might that work link to other places, et cetera, given that I've never visited and don't know much about it. You didn't ask that. You didn't ask that part. But
I had the caveat there.
Yeah. So there it's on a lake. That's observation number one, and there are pretty impressive networks, largely driven by GLION, which looks at systematic variation in community structure, nutrient dynamics, et cetera, in lakes around the world. That's the easy answer, but the kind of work that I do, which is terrestrial ecology, in large part is based on the organisms that crawl around in leaf litter. And so there are forests there, there are management practices that have occurred there, which I think provide opportunities for large-scale experiments. So if there are monocultures of different species or stands of different ages, that allows you to ask questions about leaf litter biodiversity and how those organisms affect soil nutrient dynamics. The other thing is that goldenrod, one of the plants I showed in one slide, is everywhere. And so one of the ideas that I wanted to pursue for a long time is how what leads to changes in population level variation in goldenrod clones, and then what are the cascading consequences of that. So I want to link global change drivers to changes in population structure to cascading consequences like ANPP, insect diversity above ground and below ground. And because goldenrod occurs everywhere, I could imagine doing that work along gradients as well, but also coupling it with experiments. I'm much better at the terrestrial ideas, I will admit. Yeah.
I'm curious if you have some ideas or directions that you see as avenues for better integrating the UMBS with the natural history collection.
Yeah, lots actually. So one of the things that I remember kicking around with ideas, I remember kicking around with people previously. Oh, sorry, that was a question about how might museums and UMBS and EEB interact more tightly. One of the things we've kicked around for a long time in IGRT proposals I've written is whether biodiversity scientists should have deep knowledge of taxonomy and systematics, phylogenetics of a focal organism. And so can you imagine a graduate training program that encourages and facilitates a deep dive into a part of the collection that is then coupled with experiments in the field or collection forays across environmental gradients? And so, you know, in my own group, were I to come here, I could imagine something like that, but I can imagine small grad training programs, not necessarily a full-scale, what used to be called IGRT, but facilitating that sort of interaction with grad students and undergrads as well. I mean, I can imagine, you know, collecting trips to UMBS, coupling that with museum visits, and then thinking about trait variation in space and time, and then interactions with people who understand how you might go about reconstructing phylogenies for those taxa. That's sort of an off-the-cuff answer, but I mean, that as an ecologist who's used museum data, I would really like to talk with museum people about the kinds of questions we could ask. Yeah.
You focus appropriately on natural sciences at the biological station. Have you any thoughts about how the curriculum there might expand to include topics like environmental history, policy, the arts?
I focused on natural sciences at UMBS. This is repeating the question. Have I given much thought to other parts of the curriculum like environmental policy, environmental history, or the creative arts? Yep. I've thought a fair amount about environmental history at particular places, because I think that just like you can do a deep dive on organismal biology, natural history of a place, you can also spend time there to reconstruct its history, both its ecological history and its cultural and anthropological history. And I think that place is ripe for that. I've also thought a little bit about the arts, because I know something about artists in residence programs, and I think that having writers in residence or undergrads who are learning something about nature writing or art in the organism or art and science to explain nature, we've done things like that in the past. I have not thought as much about environmental policy at that particular place, but I think you could do that in a whole host of other things with the PITE program, or just PITE, I should say.
Why don't we thank Nate for a very interesting talk?